Hello and welcome once again to another podcast from regionalfortrainees.com. This is a new podcast series where we are starting a podcast for a couple of topics, mainly didactic lectures, and we're going to be talking about things in regional anesthesia, starting from anatomy, physiology, pharmacology, and the various techniques and things in regional anesthesia. And this one is the very first podcast of that series. Let's talk about the neuron. And before we get into any further discussions, let me just lay it out flat. This is an educational tool. It is not meant for any profit. And I've tried to use resources which are already available in our open resources. So the nervous system basically has got an afferent limb, a central processing unit, and a peripheral efferent limb. The afferent and the efferent limb are the two arms of the peripheral nervous system, whereas the processing is mainly focused in the central nervous system. And of important to note is that the afferent limb gets its afferent nerves from general somatic afferents, special sensory afferents, and general visceral afferents. And this nervous system in our body is basically subdivided or set up in a hierarchical fashion, mainly being divided into central nervous system and peripheral nervous system. Of course, the brain and the spinal cord comprising the central parts, whereas the peripheral nervous system is subdivided into somatic and autonomic nervous system. And it is the autonomic nervous system which is mainly responsible for maintaining the homeostasis in a body, containing two arms. One is a sympathetic and the parasympathetic arm. So what we are trying to focus in this podcast is trying to focus upon the basic building blocks of the central nervous system, which are the neurons. And from a physiological and anatomical estimate, there are about 100 billion neurons in the nervous system in a human body. But the glial cells, which are the supporting structures for these neurons, far outnumber the number of neurons, approximately being about 10 to 50 times that number. For a neuron, the most important property that we need to focus upon is its excitability. And it is by the virtue of this excitability that it is able to communicate with different cells in our body. Neurons are not the only excitable tissues in our body. There are other ones, and mainly exemplified by the muscles. And by excitability, by virtue of this property, they are able to generate membrane potentials. And these potentials can be generated at the receptor end, which is the most peripheral part of the afferent limb. And then these potentials are propagated centrally by virtue of an action potential, and then transmitted further on from one neuron to another by virtue of synapses, and resulting in formation of synaptic potentials. For the anesthetist, I think the most important potentials are the action potentials, because by virtue of using sodium channel blockers, what we are effectively doing is either interrupting the mechanism of formation of action potentials or interrupting the propagation of action potentials, which translates into clinical action. Let's have a look at the structure of a neuron. A neuron basically comprises of its afferent part, which is the dendrites. They converge upon the cell body, which is also known as soma. The soma subsequently gives out an axon hillock through which an axon leaves the soma. The axon may be myelinated or unmyelinated. In this case, it's a motor neuron, and hence you can see the myelination. Myelination is interrupted regularly at the node of Renvir, which is about 1 micromillimeters in its diameter, and occurs regularly at an interval of about 1 millimeter. And subsequently, the axon terminates into an axon terminal, which comprises of small knob-like structures, which are also called buttons. So in a sense, there is a dendritic zone, then there is an initial segment which gives out the axonal process, and finally the axon terminates by arborizing into various nerve endings. And this can be seen here. And the various button-like nerve endings maybe hundreds of small arborization, which then subsequently converge upon the cell body of another neuron, as can be seen in the shown diagram. So what do we need to know? We need to know what are the type of neurons, something about myelin, something about the plasma membrane, something about axonal transport, the synapses, resting membrane potentials, action potentials, neuronal processing, different types of nerve fibers, and finally, how are these nerve fibers arranged in a given nerve fiber bundle. So let's begin with discussing what are the types of neurons. Neurons can be classified either anatomically or functionally or physiologically and so on in different ways. One of the most common ways of classifying neurons is anatomically by having a look at their axons. In vertebrate organisms, the axons are generally either would have more than one axon. Unipolar axons are generally seen in invertebrate organisms. In case of vertebrates, you would see the most common neurons being multipolar, which have more than one axon, exemplified by the motor neuron in the spinal cord. Bipolar neurons, which are seen in the bipolar cell layer of the retina, and pseudo-unipolar, which basically means there is a single axon, but it gives two branches which goes in two opposite directions. This is exemplified by the ganglion cell in dorsal nerve root ganglion. Apart from that, depending upon myelination of a neuron, they can be classified as myelinated or unmyelinated. So the myelin basically is a lipid protein complex that encircles a given axon. In the peripheral nervous system, it is formed by the Schwann cells, and in the central nervous system, it is formed by the oligodendrocytes, which may result in myelin formation for more than one axons, as can be seen in the lower diagram. And this one shows the electron microscopic picture of myelins and its different layers surrounding a given axon. Apart from that, neurons can be classified depending upon the functionality into a sensory, motor, or autonomic. They can be classified into which direction they carry the impulses, whether they bring in the impulses or they take the impulses out, and so on. They may be classified upon their nerve characteristics, their conduction velocities, presence and absence of myelin, their thickness, and so on. And finally, they may also be classified depending upon the type of neurotransmitters they release at the synaptic junction. When we talk about myelin, we need to know that this is a lipid protein complex. Its basic function is to act as an insulator. Now, by virtue of acting as an insulator, it results in what we call as saltatory conduction, which basically comes from the Latin word saltare, which translates into to hop or to leap. Basically, this is a type of propagation mechanism in an action potential by which the depolarization changes which are happening on the surface or the membrane of the axon jump from one node of Renwi to the next node of Renwi, and this improves the conduction velocity but eliminates the need to increase the diameter of an axon. Basically, the conduction velocity in any given axon is proportional to its diameter. So if you've got a thicker axon, you would expect its conduction velocity to be higher. If you've got a smaller or a thinner axon, then you expect its conduction velocity to be lower. But if you add myelin to it, then without the need for increasing the diameter, you can still achieve high conduction velocity by the virtue of saltatory conduction. And this is what we see in myelinated neurons, which is also responsible for the grey matter formation in the central nervous system. The peripheral nervous system, the myelin is formed by Schwann cells, and in the central nervous system it is formed by the oligodendrocytes. We've already touched upon those. What happens in demyelination disorders is that there is a loss of myelin sheath, and therefore either you got reduced conduction velocity or you have reduced amplitude of the action potential. And this can be seen in multiple sclerosis, transverse myelitis, Guillain-Barré syndrome, and so on. Histologically, what you see is this kind of a picture, which is showing an electron microscopic picture of demyelination. The first arrow depicts the axon of a given neuron, and the second arrow now depicts the Schwann cell, which is encircling the axon, and the third arrow depicts the periaxonal loss of myelination. Next, we need to know something about the basic structure of the plasma membrane. Any plasma membrane in vertebrates, or in fact invertebrates, comprises of two lipid layers. Therefore, it is a lipid bilayer, and these lipids are phospholipids. And it is the phosphorus part which gives it the hydrophilicity, and therefore the hydrophilic heads of each lipid bilayer face outwards. So the upper lipid layer, the hydrophilic part is facing outside to the cell membrane, whereas in the lower layer, the hydrophilic part is facing inside the cell membrane. And the hydrophobic tail ends, which comprise of the hydrophobic chains, face each other, resulting in formation of some sort of a bilipid sandwich, so as to speak. Apart from that, there are several proteins which can be seen transversing the lipid bilayer. They may be either integral proteins, which is that they cross only the outside layer or the inside layer, or they may be transmembranous protein, which is that they jut out of both the inner and the outer layers, as can be seen here. Some of the proteins may have carbohydrate side chains and therefore result in formation of glycoproteins and play different roles. Of importance and of note, different proteins can form either pores or channels which may allow different substances to pass through the cell membrane, or they may have particular ligands. That is, when a particular ligand comes and attaches to that particular protein, the pore channels open up and results in passage of a given substance through the cell membrane. Or it can also be voltage gated, that is, depending upon the polarity of the cell membrane, they may open and close and so on. Sodium channel is one of the transmembranous protein which is voltage gated. It constitutes of an alpha and a beta subunit, the alpha subunit being the most important. The alpha subunit consists of 4 different parts, 1, 2, 3 and 4, which are basically encircled as shown on the right side of the diagram, and each domain then consists of each parts then consists of 6 membrane spanning domains which play different role. We would be doing a separate podcast on the sodium channel and its functioning, so I wouldn't take too much time on this, but we need to remember that there are voltage gated channels on the plasma membrane, mainly being the sodium, which is where we need to focus in a neuron. Obviously, it's not possible to have an action potential without either having potassium channels or chloride channels or calcium channels and so on. So those are also important channels to remember, but most important is the sodium channel. Next, coming to an axon, which is a finer cable-like projection coming from the cell body. It may be myelinated or unmyelinated, important function being carrying nerve impulses. It specifically has a very high concentration of sodium channels, which is obvious because of the propagation of action potential, and importantly transports substances either downward direction, which is called the orthograde, from soma to the axon terminal, and these are the proteins and the polypeptides which are important for formation of neurotransmitters, or the transport can be retrograde. In this case, some of the neurotransmitters which are absorbed from the synaptic terminal are sent back to the soma for reprocessing, for recycling. Sometimes they can transmit viruses, sometimes they get hold of nerve growth factors and transmit it across in a retrograde manner back to the cell soma. And then the axon arborizes into small knob-like structures or buttons, which eventually common interface with the receptor site result in formation of an interface called a synapse. The synapse essentially consists of presynaptic knob, followed by the synaptic gap, and followed by the postsynaptic membrane. And it has got a particular physiology that needs to be understood. We would try and incorporate this physiology in some other podcasts. For the minute, this is what we need to remember. Importantly, synapse can be classified depending upon its anatomical or functional characteristics into different types. And if an axon terminates upon a dendrite, then it is called axodendritic synapse. If an axon terminates upon an axon, it's called axoaxonal. If an axon terminates on a cell body, then it is called axosomatic. And if a dendrite terminates on a dendrite, then it's called dendrodendritic. Functionally, the synapse can be formed either by incorporating release of neurotransmitters, which are contained in vesicles, and hence this type of synapse is called vesicular. Or sometimes they can be closely associated with each other without release of any vesicle, and the information passes on from one side to the other through existence of gap channels, which basically allow passage of small ions. Neurophysiology is an important cog in the wheel. It was through the suggestions of John Zachary Young, who was a UK-based neurophysiologist from 1930s and 1950s, who suggested that by studying giant squid neuron, we could understand more about the physiology of a neuron. And what he suggested was that we could insert the electrodes into a squid axon, because the axon of a squid is fairly large, 1 millimeter in size, and the techniques that were used in the 1930s and 50s were not as advanced. So we basically needed a large axon to study with the primitive technologies that we had at that point of time. And he was the one who initially suggested that we can study the neural mechanisms by using axons from a giant squid. But it was the work of Hodgkin, Huxley and Eccles which translated his suggestion into practicality, and they were the one who actually conducted a lot of work. So John Carew Eccles also worked upon synapse and its transmission, and for this wonderful work they received Nobel Prize in Medicine and Physiology in the year 1963. And what they basically found was that they took giant squid axon, put electrodes, one electrode outside the axon membrane, one electrode inside it, and as soon as they did this, they could see a potential difference on the galvanometer, and this potential difference was called the resting membrane potential. It was obviously later found out that the resting membrane potential occurs because of the difference in the ion, in the potassium conductance across the membrane, and it is the potassium channels that are responsible for formation of resting membrane potential, which is generally about minus 70 millivolts in case of a neuron. Upon the stimulation of such a neuron, they were able to see that there's a change or a perturbation in the potential difference that occurs across a resting membrane potential. And when it was of a particular value, it could be propagated. That is, it was called an action potential because there was some motion in it, which is implied by the word action. And they were able to see the shape of this action potential by employing cathode ray oscilloscope. And they eventually found that it was because of sodium, because when they decreased the sodium concentration outside the neuron, they saw that the height of this action potential started decreasing, and therefore they could indirectly infer that it must have something to do with the sodium channels. And the initial stages, these studies were basically based by altering the concentration of ions either outside or inside. Later on, as the electrophysiology techniques evolved, and especially the voltage clamping techniques came into being, they were able to alter the voltage itself from within the neuron, and therefore able to study the hyperpolarization and depolarization of the neuron membrane even better. So from the resting membrane potential, when the potential approaches zero, then that particular inward flowing current is termed depolarization. However, as positive charge moves outside, you find that the resting membrane potential becomes further negative, and a hyperpolarization is said to occur. Repolarization is a phenomena of approaching the resting membrane potential after a given depolarization. So these are the terms that we need to understand. Not all depolarization would result in formation of action potential. Only those depolarizations potentials which reach the threshold potential, here shown as minus 55 millivolts, would be eventually resulting into formation of a propagated action potential. So action potential is a short-lasting propagated event in which the electrical membrane potential of a cell rapidly rises and then falls down. And it is a property seen in excitable tissues. It is one of the ways of passing information from one cell to another. It doesn't take place unless the depolarization potential exceeds the threshold potential, here again shown as minus 55 millivolts. There can be subthreshold potentials which are called electrotonic potentials, but obviously do not result in action potential. And the given stimulus for that would result in action potential should be rapid. And if that doesn't happen, the cell membrane quickly adapts and the depolarization quickly dies down. This particular phenomena is quite important even in using peripheral nerve stimulation, because if you notice that in peripheral nerve stimulation, the current used is a square wave form. And the reason why a square wave form is used, because you want the current to suddenly rise quickly to a level where it can potentially cause depolarization. If you use a sine wave pattern of stimulation, then because the current rises slowly, the nerve membrane would quickly get adapted, and you might not get depolarization. So that's the reason why we use a square wave pulse in peripheral nerve stimulation. And the last property of action potential is that it is either an all or none phenomena. That is, if the given electrical stimulation or chemical stimulation reaches the threshold potential, then action potential will happen. And if it does not reach the threshold potential, then it will not happen. So either it is a complete or not at all kind of a phenomena. That is all or none law. Finally, what is also important to realize is that the processing unit in the brain and spinal cord makes use of different types of patterns, different types of circuits. So either you can have divergence, that is, an information from one given cell can move down to different neurons, resulting in a widespread action and so on. Or it can lead in convergence, that is, a singular cell is getting input from various different neurons. It can also involve serial processing one after the other. It can involve parallel processing, and it can involve reverberation circuits as well. So these are different physiological patterns of neural processing, and each one has got different applications as such. Nerve fibers have been classified by Erlanger and Gasser into different groups: the A fibers, the B fibers, and the C fibers. Generally, the A fibers are the thicker fibers, so their diameters are slightly thick. C fibers are the thinnest of the fibers, and the diameters are fairly thin. And B fibers are the ones which are intermediate. And different type of nerve fibers have got different properties. you can expect that the thickest nerve fiber would need to be the one who would need to have the maximum conduction velocity. And not surprisingly, you find that the thickest nerve fiber, which is A alpha, has got the highest conduction velocity. And interestingly, you see that the myelin content is also the highest. And these are exemplified by the motor nerve fibers. The C fibers, which are the thinnest nerve fibers, also have the lowest conduction velocity, as is expected. Additionally, they also don't have any myelin content. And these are the ones which transmit pain, temperature. Sometimes they're also called the slow fibers. The B fibers are mainly autonomic preganglionic fibers, which lie intermediate between the properties of A and B. Finally, what we also need to understand is the structure of a given nerve bundle. The axons are arranged together into fascicles, which basically are bound by an endoneurium. Different fascicles then are bound together in a perineurium. And these groups of fascicles lie together in a given nerve bundle surrounded by an epineurium. And each epineurium is also supplied by arteries and veins, as can be seen in this picture. These are called the vasa nervosa. A given injection between the fascicles, which is called the interfascicular injection, may not result in injury to the given nerve fibers. And therefore, it has been seen that even with intraneuronal injections, which are interfascicular, you may not see nerve injuries. However, if an injection happens to be intrafascicular, the chances of having injury rise tremendously. And these have been one of the recent elucidations of papers. So that is it. This is the first podcast in the series, and we've discussed Neuron. If you like this, then I would encourage you to share it with your friends, either on Facebook, Google, or through Skibid. You can also subscribe to my channel, which is hermansehmbi 29. That is it, folks. Until we see you next time around, thank you so much for watching this. This is regionalfortrainees.com. Tune into learning.